Hey everyone, and welcome. You've probably done this in a biology class at some point, the classic starch test on a leaf. But that simple procedure hides some really fascinating complex chemistry. So today, we're going to go way beyond the basics and actually explore this test from a molecular perspective. Get ready to see what is really happening inside that leaf.
Okay, so before we dive in, just a quick heads up. This is going to be a much more detailed, more technical breakdown of the science behind what's going on. We're kind of assuming you're already familiar with the basic steps. If you're feeling a little rusty or want a refresher, I'd really recommend checking out our first explainer on this topic. But for everyone else who's ready for the deep dive, let's get into it.
So, here's our game plan for today. We're going to start with the key molecules involved, then we'll deconstruct the procedure to see why each step is so important. After that, we'll get into the cool part, the molecular reaction that causes that famous color change. Then, we'll talk about interpreting the results, and we'll wrap up with the big scientific principles you should take away.
All right, let's get right to the chemistry. To really understand this test, we absolutely have to start with the molecules we're working with, and we'll begin with our main target, starch.
So starch isn't just one simple thing. It's actually a massive polymer, basically a super long chain of glucose molecules all linked together. You can think of it as the plant's way of banking all the energy it makes from sunlight. And this complex structure, well, it's absolutely critical to how this test works.
And even then, starch is actually a mix of two different kinds of polymers. You've got amylopectin, which is all branched and water-soluble, but the real star of our show is amylose. It's a long, straight chain that naturally twists itself into a helical coil, kind of like a tiny molecular spring. Now, I want you to remember that coiled shape. It is the absolute key to this entire reaction. And here's the first big misconception we need to clear up. We call it the iodine test, right? But it turns out pure iodine isn't actually the active ingredient here. The real work is done by a special charged ion called the triiodide complex.
So how do we get this magical complex? Well, pure iodine, the I2 molecule, doesn't dissolve in water. It's a huge problem. That's why the solution we use also contains potassium iodide, or Ki. When you dissolve Ki in water, it breaks apart into ions, and one of those is the iodide ion, I-. This little guy then grabs onto an iodide molecule, and together they form that soluble and, more importantly, reactive triiodide ion. Seriously, without potassium iodide, the test just fails.
Okay, so now that we know our key molecules, we can look at the experiment not just as a recipe to follow, but as a series of deliberate steps where each one has a very specific scientific purpose.
The very first step is probably the one you remember most clearly, putting that leaf into a beaker of boiling water.
But this isn't just about softening it, no. Boiling does four really critical things. First, it kills the leaf tissue and completely denatures its enzymes, which stops all biological activity instantly. Second, it starts to break down that tough, waxy outer layer. Third, it disrupts the cell membranes and even the rigid cell walls. And all of that together achieves the main goal. It makes the leaf permeable, like a sponge, for everything that's coming next.
Next up, the leaf gets moved from the boiling water into a bath of hot ethanol.
So why ethanol? It's a great question. It's because ethanol is a solvent that can do something water can't. Chlorophyll, the molecule that makes leaves green, dissolves perfectly in ethanol. This whole step is really just for visual clarity. If we left the leaf green, that intense pigment would totally hide the blue-black color we're hoping to see. And of course, a quick safety note, since ethanol is super flammable, it's always heated in directly in a hot water bath, never over a direct flame.
After the ethanol bath, there's one last little prep step, a quick rinse in some warm water.
Now this might seem kinda minor, but trust me, it's crucial for actually handling the leaf. That ethanol treatment leaves the tissue incredibly brittle and crispy. If you try to flatten it out, it would just shatter. Dipping it back in water softens it right up, letting you spread it out on a tile without it breaking into a million pieces.
Okay, so our leaf is now killed, it's permeable, and it's been decolorized. It is perfectly prepped. So let's move on to the main event, the molecular reaction that produces that famous, dramatic color change.
This is where it all comes together. Remember that helical coil of amylose we talked about? And that long, thin triiodide ion? Well, the reaction happens when that ion slips perfectly inside the amylose coil. It's a perfect fit. This creates a brand new structure called a charge transfer complex, which is just a fancy way of saying the molecules are now so close that electrons can move between them in a whole new way. And this new complex has a very unique property. It is incredibly good at absorbing visible light. When light hits it, the complex basically soaks up almost all of the colors, reflecting back only the wavelengths that our eyes see as a deep, intense blue-black. So it's not a stain, it's not a dye, it's a physical change in structure causing a change in how light behaves. Pretty cool, right? Now that we know exactly how that color gets formed, let's connect that chemical reaction back to the bigger biological picture. What are we actually learning when we see that final result?
It's really a simple binary outcome. If the leaf turns that dark blue-black, we know for a fact that the triiodide amylose complex is formed. Chemically, that means starch is present. Biologically, that's our evidence that the plant has been performing photosynthesis. On the other hand, if the leaf just stays that yellowish-orange-brown color of the iodine solution, it means no complex formed, there's no starch, and therefore either photosynthesis didn't happen or the plant already used up all its starch reserves.
This is such a classic and powerful demonstration. But like any scientific test, it's so important to understand what it can tell us, and just as importantly, what it can't tell us. We need to know its applications and its limitations.
The applications are fantastic, especially for teaching. It's the absolute go-to method for visually demonstrating that photosynthesis is happening. You can also use it to investigate how things like light or carbon dioxide affect starch storage. Or you can do that amazing experiment with a variegated leaf to prove that chlorophyll is necessary.
But you have to understand the limitations. First, this is a qualitative test. It gives you a yes or no answer for starch, but it can't tell you how much starch there is. Second, and this is so important, a negative result, no blue-black color, doesn't prove photosynthesis didn't happen. It only proves there's no stored starch. The plant could be making glucose like crazy and just using it all up for energy right away instead of storing it. So let's wrap all this up by summarizing the core scientific principles we've covered today. If you take anything away from this deep dive, it should really be these 5 key points.
First, starch is just stored energy. Second, that reaction only works because of amylose's unique coiled shape. Third, the true hero of the reaction is the triiodine ion, not pure iodine. Fourth, every single step in the procedure, the boiling, the ethanol, has a very specific and necessary purpose. And finally, number 5, that blue-black color isn't a stain. It's a whole new molecular structure, that charge transfer complex, that's changing how light itself is absorbed.
And that brings us to the end. We've seen exactly how this test works on a deep molecular level. So I'll leave you with a final thought to chew on. This test is amazing at showing us where starch is, but what are all the different environmental factors, beyond just an obvious lack of light, that could prevent a plant from being able to make and store that energy in the first place? It's something to think about. Thanks so much for joining me.